Hello, I'm Eric Vaskovich. I'm the lead bioinformatician for cancer services at the All Wales Medical Genomics Service. Croeso, welcome. Actually, one second, my slides aren't working. There we go. Okay, so Croeso, welcome. So I'm joined today by Laura and Joseph, two other bioinformaticians at the All Wales Medical Genomics Service. And together we're going to give you an overview of our service and some of the developments we've been involved in. So to start with, I'm going to go just an intro to AWMGS and our team and the wider genomic strategy within Wales. And then I'm going to hand over to Laura, who's going to talk about some of our updates from the cancer section, and Joseph, who's going to talk about updates from the constitutional section. So yeah, an intro to AWMGS and our bioinformatics team. So AWMGS is a NHS genetic diagnostic service. So we provide genetic testing for patients across Wales. And we're an ISO accredited lab, and we represent the clinical and laboratory services for the lab. There we go. So we're based in Cardiff, in the area there, in the University Hospital of Wales. So this is the largest hospital within Wales, but we serve the whole of Wales for genetic testing. So there's 7 health boards within Wales shown on the map, and they all send their samples down to us where we do the processing. So just a quick overview of some of the services we provide within the laboratory. So over on the left, we've got things that, as a bioinformatician, I'm not particularly clued up on, but we've got some cytogenetic techniques, so things like G-banding and FISH, genotyping, so a range of PCR tests for a range of different services. And some high throughput sort of automated services. So we've got automated DNA extraction, PCR, and Sanger, etc. We've got a range of cell-free DNA services. So we've got a droplet digital PCR service for ctDNA, and also an NIPT service. And then over to the right, we've got a range of NGS sequencers where we run a variety of panels, and we've got a team of bioinformaticians and clinical scientists who are involved in analysing this data. On the clinical side of the service, we've got a team of genetic counsellors and clinicians who are involved in counselling and sort of providing specialist clinical support to patients across Wales. So as the 3 of us are part of the bioinformatics team, I'll give us a quick intro. So currently we've got 5 members of the team, so 3 of us are here today. And our team is expanding, so we've recently put 2 job adverts, so we should have new stuff soon. And also just a quick sort of advertisement, we have an SCP position that will be advertised soon. Please ask in our booth if you want some details on this. The application process is different to England, but it will be on the same SCP curriculum as England. So please ask us about this if you're interested. So our team are involved in various projects across the lab, and we have access to cutting-edge sequencing and compute resources. So this is just a quick schematic of the resources we have access to. So we've got a range of sequencers. So we have a NovaSeq, where we run a whole genome sequencing service that Joseph's going to talk about. We've got a couple of NextSeqs, MySeqs, and HiSeqs as well, which are used for a variety of different panels, depending on how much sequencing we need. All of these sequencers save the data to our REN HPC system. So this is hosted by Cardiff University ARCA system, and they take care of all our sysadmins, so we can just worry about producing pipelines and generating data. As well as this, we've got a couple of trading servers that are plugged into the REN system. So this is used for rapid processing of large amounts of data, so particularly for rapid whole genome sequencing. And then we've also got a range of clinical interpretation and then software that interacts with this as well. So that's a quick overview of AWMGS, and the bioinformatics team. I'm just going to do a quick run through of the wider strategy within Wales. So this is a picture of Vaughan Gethin, our health minister, and this is a quote from back in 2017 when he launched the Genomics for Precision Medicine strategy within Wales. As part of the genomics strategy, sorry, I've gone too far now. As part of the genomics strategy, the Genomics Partnership Wales was set up, and this is essentially a umbrella organisation that's sort of trying to bring together all the various partners involved in genomics across Wales. And their sort of mission statement is working together to harness the potential of genomics to improve the health, well-being and prosperity of the people of Wales. So Genomics Partnership Wales, or GPW, is split into various strategy delivery work streams, and these are essentially different working groups for that with the aim of delivering the strategy. So we've got a variety of things, from workforce and training to IT infrastructure, commissioning, bringing together research and innovation, communication, patient-public involvement, etc. So on the right, we've got a picture of the different partners in GPW. So we've got partners from the NHS, from university and research, Welsh Government, and patients and public. And the aim really is to drive collaboration across research and the private sector, and improve genomics within Wales. From a bioinformatics perspective, it sort of gives us access to a larger bioinformatics community. So the two circled ones there, we've got the Pathogen Genomics Unit, or PENGU, who are based upstairs from us, and they do pathogen bioinformatics, and they've been doing some really interesting stuff during COVID. And we've also got Wales Gene Park, who are involved in research bioinformatics, so sort of bridging the gap between research and clinical. So I'm sure we'll have someone from GPW that will be available at some point for discussion in our virtual booth if you'd like to know more about that.
8 minutes, go
on. So just to wrap up my bit of the talk, so the future plans really are to build upon what GPW have already done, and just to establish a precision medicine centre for excellence within South Wales. And our Managing Director, Clive Morgan, has recently been appointed an Innovation Fellow at Cardiff University. And a key part of this is to, so we've outgrown our current building, and we'd like to co-locate with other genomic services within a new site within the not so distant future. So I'm going to pass over to Laura, who's going to talk about implementing some new cancer services at AWNGS. So, thanks.
Okay, so hi everyone, I'm Laura. So I'm one of the bioinformaticians as well at Wales Medical Genetics Service, and I'm going to talk about implementing new cancer services. That one. So first I'm going to talk about the fusion testing service. So this started due to NICE guidelines, which showed that we should be offering a drug called larotrectinib after testing for fusions within the NTRK genes. However, as part of this service, we also decided it would be good to offer testing for fusions within other genes that we know are involved in solid tumours. So if you look at the graph on the right, you can see that in July 2020, we were predicted to get about 10 samples a month, which meant that FISH was a very viable option. However, after October 2020, we were predicted to get 100 samples a month, which would then be increased to 400 in January 2021. Which meant that FISH was no longer a good option and would have to switch to using an NGS pipeline. So my job as a bioinformatician on the project was to design this pipeline. So I started looking at some of the different fusion detection software that's available. So a lot of the fusion detection software work by looking at the number of split and spanning fragments. So the split fragments are the ones where the read is split and a part of it aligns to, for instance, one gene and another part to another gene. You also have the spanning fragments, which is where you have the paired end reads, and one of the reads aligns to one of the genes and another of the reads aligns to a lot further away than you'd expect paired end reads to. So after looking through the literature and trying out different software, the best ones that worked with our data was the Star Fusion and Ariba softwares, as these worked by detecting the spanning and split reads, like I just described, as well as applying numerous different filters to remove the false positives. Whilst these two pieces of software were really good, they weren't able to detect the intergenic fusions, or exon skipping events, as they're also called, So for this, we used a tool that's designed to detect alternative splicing events called RMATs. So the combination of these three software worked really well. And alongside applying some thresholds, such as to the PCR qubit value, the number of aligned reads, and only using samples with a concentration greater than 50 nanograms, we managed to get a sensitivity of 99%, a specificity of 100%, and a pass rate of 83%. Whilst this pass rate was not as high as we originally liked, due to we're used to dealing with DNA, we realized that for RNA, this was actually good and enabled us to go live with the service. So we're now testing for fusions for our patients. So the second service I want to talk about is the DPYD service. So if you look at the diagram on the right, just to give you a bit of background, the DPYD gene codes for the DPD enzyme, which then breaks up 5-fluorouracil to dihydrofluorouracil. So if patients who have solid tumors are offered fluoropyrimidine-based chemotherapies, so if patients have certain variants within the DPYD gene, this can lead to decreased function of the DPD enzyme, meaning that there can be a buildup of this 5-fluorouracil, which can lead to toxicity, which can be fatal. So we, as part of the DPYD service, we test for germline variants in the DPYD gene to enable us to adapt the dosage of the chemotherapy that we give to patients. So the genotyping assay we use looks for these 5 different variants, which were found using the 100,000 Genomes Project. If you look at the table, you can see that the top 2 variants are predicted to give no function to the DPD enzyme, whereas the final 3 variants are predicted to decrease the function. So depending on the combination of the variants that the patients have, they can be given a different dose of the chemotherapy. So the clinicians use the CPIC guidelines, so who categorize patients into 3 different groups. So you have your normal metabolizers who have 2 wild types and can be given the normal dosage of the treatment. You have the poor metabolizers who have either 2 variants which are predicted to have no function, or no function variant and a decreased function variant, and they're given, it's suggested not to give these patients this type of chemotherapy. And then you have the intermediate metabolizers who can be given a reduced dose of the chemotherapy, and then this can be either increased or decreased depending on how they react to the treatment. Okay. So the future developments that we're looking at working on in the lab are somatic CNVs. So we're planning on creating an NGS pipeline to detect somatic CNVs instead of FISH in a similar way we did with the fusions. We've also got projects going on looking at tumor mutational burden, microsatellite instability, and ctDNA. So I just want to thank everyone who is involved in the 2 projects. And I'm now going to pass over to Joseph, who's going to talk to you more about the WINGS project. Thank you.
Hello, everyone. I'm Joseph Halstead, and I'm the lead bioinformatician for constitutional services at Orwell's Medical Genomics Service. So today, rather than going over the constitutional service in general, I'm going to focus on one of our kind of flagship services which we developed in the last year. And this service is known as WINGS. So WINGS stands for Wales Infant and Children's Genomics Service, which is a bit of a forced acronym, but it works. And it's a rapid whole genome sequencing service for critically ill pediatrics. And basically the purpose of this test is designed to reduce patients' diagnostic odyssey. So these children which are born very sick, you don't have to wait years to get a diagnosis. We can get a diagnosis straight away and hopefully find something where we can intervene and help that child's prognosis. So it was launched last year in April under quite challenging conditions due to the COVID pandemic. So we're quite proud that we managed to push the project forward during those circumstances. And basically, we sequence the proband and the parents on a NovaSeq at 2 times 150 reads. And we have a reporting time of less than 14 calendar days. So we're hoping to get this down, but because we didn't know how long it would take essentially to report some of these cases, we kind of overestimated the reporting time at first, but we have reported them much quicker than that. So this is really quite an innovative and world-leading service. So we were the first in the UK to introduce a rapid whole genome sequencing service for critical pediatrics, and I think it really showcases the genomics and bioinformatics talent that we've managed to build up here in Wales. So doing clinical whole genome sequencing is pretty hard, even when you have a long time to do reporting. So rapid whole genome sequencing raises its own specific challenges, which we had to overcome. So the next part of the presentation is going to focus on how we overcame those particular challenges. And the first one of those was how to actually align the reads and variant call in quick enough time. So we used, as Eric mentioned earlier, we've got a Dragon service. So we used that for alignment, duplicate marking, QC metric generation, and variant calling. So we do this per sample, and then we have one final step, which is the joint calling of samples within each run. This allows us to call de novo variants much more accurately. And we stuck with genome build 37, although 38 might have been a better option. We thought because we wanted to move fast with this, we didn't have the time necessarily to validate against 38 and change all our downstream tools to 38. So we stuck with 37, but moving to 38, as with a lot of clinical genetics laboratories at the moment, is a future goal, which we'll probably implement soon. And so initially this service launched, we just called SNPs and indels using the standard variant caller on the Dragon, but we also call CVs and structural variants using depth-based and split read variant callers. But we haven't validated the sensitivity of these, so they're not part of the accredited test at the moment, although we hope to change that soon. So to design this pipeline, we kind of split it into 2 bits. So the first part runs on the Dragon, and the second part runs on our standard high-performance computer. So the first part does the variant calling, and the second part does the annotation, QC metric generation, and variant filtering. And we found splitting the pipeline in 2 like this really helped us move a lot faster, because we could develop the bits of the software that we needed on the high performance computer, which is a lot simpler than doing it on the Dragon. So the next challenge we had to overcome was how to filter the variants, and how do we find clinically relevant variants in a short period of time. So we developed a versatile Python package known as PyVariant Filter for conducting family-based analysis on NGS data. So this package can be used to design workflows for clinical and research, and it allows bioinformaticians to quickly test and develop and then validate new variant filtering workflows and this kind of data. And it's really fast too, which is a strong requirement for rapid whole genome sequencing. So we can filter whole genome samples in about 10 minutes, and we can do WES samples in a few seconds. So again, this showcases that we can rapidly and safely develop bioinformatics software for quite complex requirements here. So what criteria did we actually use to filter the variants? So we used a few different filtering criteria to really kind of up the amount of variants that we could find. So to find deep intronic variants, which are predicted to impact splicing, we used Splice AI, and we can whitelist any variants which have a Splice AI value of more than 0.2. We exclude any variants which have a nomad frequency more than 1%. And then we also do inheritance-based filtering. So we look at de novo, autosomal recessive, compound het, X-linked recessives variants, or variants that have been inherited in a UPD manner. And then we also whitelist ClinVar pathogenic variants, which are low frequency. And then to minimize the number of variants we look at, we initially look at a panel of about 4,000 genes. And then if we don't find a clinically interesting variant in those 4,000 genes, we expand the panel to look at all genes. And this allows us to move through the cases a lot faster. And also we look at certain vet consequences, as you can see on the right here of the slide. So we look at frame shift variants, missense variants. So how do we validate this test to make sure that it was fit for purpose? So we did quite a robust validation here at All Wales Medical Genomics Service. So we processed multiple runs containing genome and bottle samples and measured the sensitivity and recall using a program called hap.py. We found that the DRAG produced excellent results on this. So we had a sensitivity for SNP of 0.999 and indel of 0.984. And then we also ran previous samples with known results from our clinical exome experiments. And to really validate our filtering criteria, we also spiked in variants into the VCF, and that allowed us to get a much more robust validation of our filtering pipeline. So I think I just skipped a slide there, but I will continue on this one. Oh, if I get back. Oh, here we go. So cases to date. So we've had 18 patients referred to WINGS. This slide's actually a bit out of date. We've had a few more since then. We've had one of these patients got diagnosed by array CGH, so we also do that in parallel at the moment until we validate in CNVs. We had one fail due to non-paternity as well. So we managed to get a diagnostic yield of about 44%, which is kind of in line with what we'd expect from this kind of sequencing experiment. And you can see we've got a variety of different variant types and a variety of different inheritance mechanisms. So we've got some de novo, we've got some homozygous variants in consanguineous families, and we've got some compound pets as well. And then we also had some variants which are inherited from the parent, and we used the ClinVar whitelist to find these. So what's next for this service? So we're going to switch to a PCR-free kit soon, which will allow us to call repeat expansions and better call CNVs and structural variants. We're going to validate mitochondrial calling. We're going to switch to version 38, and we've got a new variant database in development, which is going to allow us to kind of keep a record of every variant that we've seen, so we can calculate in-house frequencies and get rid of artifacts and generate in-house whitelists, that kind of thing. And then we also want to use this tool that's been built at Imperial called UTR annotators to find possibly pathogenic 5' UTR variants, and that's also been incorporated in the next release. So, any questions? That includes Eric and Laura's talks as well. So if you've got any questions, put them in the chat. I can see one question already. So this is obviously very focused on the incredible work All Wales Medical Genomics Service has done, but how easily can it be applied to others? So I think what they're getting at here is maybe can other people use our solutions, possibly? So I guess we're always looking for people to collaborate. So if anybody has any potential collaborations, then please come and speak to us in the booth, and we'd be happy to chat to you. And a lot of our software is open source, so we can share that code. Cool. We're All Wales Medical Genomics Service. Thanks for listening and watching. Goodbye.
We have a booth, so if you come and visit us at the booth, then we can answer any more of your questions, or you can see some more of the work that we've been doing. So, yeah, thank you very much.
Thank you.